Okay, so we are going to start Tokyo Nodai 130th Anniversary Symposium, Nutrition and the SDGs. For the opening, our department dean, Dr. Yuji Yamamoto, will present. Dr. Yamamoto.
So good morning and good evening, everyone. My name is Yuji Yamamoto, and I'm very pleased that we can hold a joint symposium with the Pennsylvania State University as the commemorate of the 130th anniversary of the founding of Tokyo University of Agriculture, Tokyo Nodai. I really appreciate staffs and students from Penn State are participating, even though it is late there in State College. Besides that, we have near 100 people who are attending this seminar, and I am delighted that many people are interested in this event. Before we start the seminar, I would like to briefly introduce about our faculty. Our faculty is consisted from 4 departments: Department of Agricultural Chemistry, Fermentation Science, Nutritional Science, and Food Safety and Nutritional Science. We all have the common interest in food, nutrition, and related fields, such as environmental science. We have a mission to educate students and build up a new concept in these fields. One of the reasons I came up to this idea to share this time with Penn State is that I thought we both have common interests, especially in the nutritional fields. And Penn State has a very strong research background in the nutritional field. As we all know, the WHO is predicting a food crisis in year 2030. So if we can share ideas, materials, if we can conduct a joint collaboration research project, if our students can learn from Penn State, if the students from Penn State can learn from our university, Nodai, I believe we can contribute to avoid these crises. We have programs which students from abroad can learn from a short and a long period of time in Tokyo. The students from Penn State can explore authentic Japanese foods here in Japan. We also have many alumni in the sake brewery field, so we can go to the brewery and visit them. Vice versa, we can send students to Penn State also. So finally, I would like to thank all the members from Nodai, and in particular, Professor Ross, for helping us in organizing this symposium. I hope this will be the first step for a fruitful exchange between the 2 universities, and we hope we can meet in face in the new future. So I don't want to be too long, and I'll pass the microphone to you, Ken. Thank you very much.
Thank you, Dr. Yamamoto. Hi, my name is Ken Iwatsuki, a professor in Tokyo University of Agriculture, Nodai. We call Nodai as abbreviation. So today, I'm so excited about this symposium, but before the first talk, I would like to show how to make a comment. As you know, if you go to the lower screen, there is a chat button. You can comment here or ask question with this chat, okay? And Japanese, for the Japanese student who is not good at English, you can write in Japanese, and I will try to translate. So I guess it's time to start. Is everyone ready? We would like to move on to the second speakers. Dr. Yaki Lee, are you ready? Sorry for this. Maybe you're not ready yet, but can you
That's fine.
Okay, good.
I can take over.
Okay, so Yaki Lee was a student of UPenn. And her talk is my research experience in mathematical modeling at Penn State. Okay, Penn State, sorry. Please.
That's fine. All right, so let me share my screen first. So can everyone see my screen now?
Yes.
Okay. So good morning, everyone, and good afternoon, good evening, everyone at Penn State. Konnichiwa. Yeah. So the topic of my talk today is my research experience in mathematical modeling at Penn State. So just a brief introduction about myself. I got my bachelor degree in biology in China at Zhejiang University. And actually, before I went to Penn State for graduate school, I did a summer intern at Todai University. And I did my PhD with Dr. Ross in nutritional sciences and did 2 years postdoc with her and then moved to National Institute of Health. Basically, during my graduate study and the postdoc, I studied vitamin A. And so a brief introduction about the vitamin A. So vitamin A is essential micronutrients which we ingested through our diet and get absorbed in our intestine, transferred to the liver, where it can be loaded or bonded to its binding protein and get secreted into the circulation, where it can be delivered or travel through our body and delivered to the target tissues, including but not limited to like brain, the adipose tissue, muscles, and therefore it has a variety of functions in different biological processes. And given its importance and also the complexity of its metabolism, it's really difficult and challenging to fully understand some mechanistics and also investigate the vitamin A metabolism. Therefore, that's where the mathematical modeling, also known as the compartment model, can come into play. So for the mathematical modeling here, I present it's a classical model of the vitamin A, whole body vitamin A metabolism. So all these circles are the compartments where we can consider them as the tissues or organs that share a similar characteristics regarding the vitamin A metabolism. And the arrows indicate the transfer or exchange of vitamin A between those compartments. And depending on our research needs, we can either expand this model, means adding more compartment, or reduce the number of models. Therefore, this flexibility of this model-based compartment analysis, allowing us to investigate the vitamin A metabolism under various physiological conditions. For example, by different nutrient status, like the nutrients deficiency, or by the different physiological status, for example, the pregnancy, lactations, or different life stages, the neonatal period. And here are a couple examples that the projects I have done with Dr. Ross regarding the vitamin A metabolism under those different status. So today, because of the limit of time, I'll just briefly talk about one of the projects, which is the impact of iron repletion on vitamin A metabolism and kinetics in iron deficient rats. So basically in this study, we have our rats in fed an iron deficient diet to develop iron deficiency. And then we gave them the vitamin A dose, which labeled with tracer and then that we can track through the blood collection during this study period. And in the middle of the study, which is the day 21, after we gave the vitamin A tracer, we have half of the rats switch to the iron repletion diet. And therefore, you can tell from the data here, the triangle are those rats continuously on the iron deficient diet, but those ones with open circles are at the middle of the study, we switched the diet. And by first of all, comparing these samples, we can tell, okay, there are some difference in the vitamin A kinetics, but what are those things? And that's why we use the compartment model by fitting those data, and that's where the lines or the dashed lines indicate. By fitting this data, we can attribute those changes we observed in these data points, these different the pattern of the kinetics, that's during the iron deficiency, the vitamin A mobilization from liver, which is this compartment in our model, may be decreased. And by recover these rats with the iron repletion diet, the mobilization of the vitamin A from the liver can increase, as well as the vitamin utilization. And that's why from this data here, we can see the increase in those data points in those iron repletion rats compared to the iron deficient rats. And this is just a very simplified explanation of this project. But more details can be found in our previous publication regarding this project or the other ones like the vitamin A metabolism during the pregnancy and lactation. And that's it. Any questions? I know I sometimes speak really quickly and may take time to respond.
Thank you, Dr. Yakiri. We will accept question or comments. Is there any question, comment?
Hi, Yaki. Yaki, is that the right pronunciation? Yaki?
Yaki or Yachi, both are fine.
Okay. Thank you for your presentation. This is Yuji. It was very interesting about you kind of focused on iron deficient. Can you briefly tell us why it was iron deficiency? Yeah.
Sorry, because of the time, I didn't provide much background information why we did the iron. So first of all, the vitamin A deficiency and the iron deficiency, they are both considered as public health issues. And one findings of this epidemiology study is that they have co-occurrence of these two types of nutrients deficiency. I mean, worldwide, that's one incidence. And the other thing is, from other study, they usually find some correlations between the vitamin A deficiency and iron deficiency. And in other prevention trials or the field study, they find if with improved iron status, sometimes they can also observe improved vitamin A status and vice versa. And that's why I want to test, okay, if there are some interactions between the metabolism of these two nutrients, what's the possible mechanism? And in our case, we first have the iron deficient model and see how the vitamin A kinetics would change if we replete these rats or replete these rats with the normal iron. And that's why we see an improvement in their vitamin A status in the plasma. And based on our modeling, we have the hypothesis this improvement is due to the improved liver mobilization of vitamin A. And this can generate new hypotheses. Okay, what's this possible mechanism or any molecular explanation of this improved mobilization of vitamin A from liver? Yeah.
Thank you, Yaki.
No problem.
So we'd like to move on to the next speakers. Carmen, are you ready?
Yes.
The next speaker will be Carmen. Her talk is about my research on heart failure at Penn State. So please, Carmen.
Hi, everyone. Yes, so my name is Carmen Annevelink. I am a third-year PhD candidate in Dr. Greg Shearer's lab, who you'll hear from later on this morning. I am in the nutrition science department at Penn State. I am in my third year, like I said, and I previously received my bachelor's from the University of California at Berkeley in biology. Thank you so much for including us tonight. So in my research projects, we hypothesize that using metabolic phenotypes and genetics, we will be able to formulate better risk predictions for heart failure. I'm approaching this project using both population level data analyses and through animal studies. First, through population studies, looking at panel A, the current thought is that within a population, there is a single distribution of metabolism based on genetics that can be influenced by diet and lifestyle to increase or reduce risk. As you can see, this green distribution can either be moved to the left by diet and lifestyle to reduce level of activity or move to the right. The diagram suggests that an entire population can be adequately described in relation to this mean. We're suggesting something more similar to panel B, where there exists more than one different metabolic phenotype within a population that are rooted in genetic differences that can be more specifically describing risk for disease. Looking at panel B, the green distribution with mean A is very close to the overall population distribution, but it doesn't encompass the genetic predisposition of each individual in that population. There is another smaller distribution in red with a different mean, described by a different genetic makeup and altered by diet and lifestyle in different ways from the green distribution. We hypothesize that these different metabolic phenotypes described by fatty acid metabolism can be used to better identify an individual's risk, specifically when their diet and lifestyle are also considered. For example, an individual from the green population with a healthy diet and lifestyle might have an index of activity of a specific enzyme that is slightly to the right of mean A, whereas an individual from the red population with an unhealthy diet and lifestyle might be shifted to the left from their genetic mean. And while these two individuals might have the same exterior metabolic activity, they should be treated differently with respect to potential diseases due to their different genetic predispositions. And so we're trying to identify the presence of these different components of the distribution. In this middle graph, I have begun to examine data from the MESA cohort. And as you can see, that this population of data from using log DHA values for these individuals, the distribution breaks into three normal components. And we found that these three components have significantly different risk for heart failure using Cox proportional hazard models. And so we're looking forward to continue these analyses in the MESA cohort and Framingham cohort and the women's health study using both phenotypic fatty acid data as well as the individual's genetic data and using Cox proportional hazard models to assess the risk. We will also continue along with these population level analyses using data from animal studies in which the animal's genetics and diet can be examined in a factorial manner to understand how oxylipids and fatty acids might change and reflect a different metabolic phenotype. And our current genes of interest include FFAR4 and FFAR1, which we are also examining in the population level data. For this part of the project, I'm excited to work with collaborators and receive those plasma samples in order to do the fatty acid and oxylipid extractions here in the lab at Penn State. With these two parts of my project, I'm definitely looking forward to how these can contribute to understanding of how to prevent diseases, but also to continue learning about heart failure and how to help people live healthier lives. So if you have any questions, let me know.
Thank you very much, Carmen. Is there any question from the audience? So I have one. What is the metabolic phenotype? Actually, is it a content of fatty acid in the blood, or what is this phenotype?
Right, yes. So metabolic phenotype is something that we are currently defining. So that makes sense that you would ask what that is. And so it is the content of fatty acids in an individual's blood, but it's also the like the whole panel of fatty acids and how the individual's metabolism is unique to themselves so that they can be used as an identifier as well as just these are the certain levels of fatty acids that they have.
Okay, so you're still in the middle of defining the phenotype.
Well, the phenotype itself will be specific to the individual, yeah.
Yeah. Okay, thank you very much. Is there any question? Okay, so we should move on because we are running out of time now. And the next speaker is Dr. Ross, but here I will switch the host to Dr. Yamamoto, who is a good friend of Dr. Ross.
Cool. Hi. Good. So I would like to introduce Dr. Ross. Dr. Ross is a professor of nutrition and physiology in Penn State. If I'm correct, this is Dorothy for her Huck chair and the department head. I won't be long, but Dr. Ross has continuously doing vitamin A metabolism research, and definitely she is one of the opinion leaders in this field. So Dr. Ross, please, can you start your seminar?
Yes, so thank you very much. Good evening, everyone. Good morning to those of you in Tokyo. So I will begin to share my screen right away. And just one moment. Let's see, share. All right. All right, well, can everyone hear me well? Oops. Great. Yeah, thank you very much, Professor Yamamoto, Professor Iwatsuki, and everyone who came to the conference today. So we're really very delighted to be able to share your celebration of the 130th year anniversary in this symposium. So since we are in a somewhat historical mood, I'd just like to start for a moment with a little bit of history about Penn State. And so here is a picture that is very famous at our university. The Penn State University was founded as one of the land-grant universities of the United States, and that happened right about the time of the Civil War. What you see in this picture, this fresco, which is a wall painting in one of the buildings at Penn State, is our President Abraham Lincoln with a student next to him and a tree, a very bare tree. And what this depicts is planting this symbolic tree, which will grow into the university of the future. So in that way, I think that Penn State University and NODI have quite a bit in common. Moving to the present, here's our building, and I think you'll see this on some other slides, and you will see that the trees have matured, and this is our research home. Chandley is the home of the Department of Nutritional Sciences, also the genomics core for the campus, and an MRI imaging facility. Okay, so I'd like to start to move into my topic, which is about nutrition and infection. And would just comment at the outset that really at no time that I can remember has public awareness of infection and vaccines been greater. So the COVID situation has really changed the world, and the public itself is very aware of immunity, vaccines, antibodies, etc. We also recognize that comorbidities are recognized as being very important for risk factors. And when you think about it, diet and other modifiable health practices are really the things that we can do to help prevent disease or reduce the severity of outcomes. So my talk this evening will be on two aspects. One, vitamin A and public health, really setting the stage. And so again, I'd like to do a little bit of historical review and then talk just very briefly about vitamin A and immunization, focusing on results from some model studies that have been conducted in my laboratory. So this is a slide that I often use when I teach to our students to point out that the vitamin A is an old vitamin and that there are early landmarks in vitamin A research that we can learn about. Vitamin A is said to have been discovered in 1913 by McCollum and Davis as an essential fat-soluble dietary component. They just called it fat-soluble A. But they recognized that it was necessary for growth and for the life or survival of animals. In Japan, Umetaro Suzuki at Riken also discovered fat-soluble A. So, very oftentimes scientists in different situations are working on similar ideas and come to discoveries very similarly. And I will show you a picture of that in just a moment. About 1925, there was a very important paper published by Wolbach and Howe in which they talked about the histopathological changes that they saw as characteristic of vitamin A deficiency. And these were changes in rats that were fed a vitamin A deficient diet. And they showed that the epithelial cells throughout the body were affected by this deficiency. Another very early paper was that by Green and Mellenby, and they described how vitamin A deficient animals in their laboratory oftentimes died of infections. These were not purposeful infections, these were environmental infections. And they talked about vitamin A as being an anti-infective agent. So before we go on, here on the next slide, I will show you your famous vitamin A researcher, Umetaro Suzuki at Riken. And I had the opportunity several years ago to visit the archives there at Riken and to see the historical presentation about Dr. Suzuki's research, as shown on the upper left. Okay, so at this time, although it was known that vitamin A was important for epithelia and it was known it was important for preventing infection, we still did not know what vitamin A was. So what is this essential factor in food? And it was discovered then in the next period that vitamin A is really retinol. Others showed that decades later, that retinoic acid, the active form of vitamin A, was characterized. And then there were important studies showing an essential role of retinaldehyde as a factor in the visual cycle. Around 1968, an important paper by Nevin Scrimshaw and co-workers, it was a monograph or a book that they wrote for the WHO. And they said that no nutritional deficiency within the animal kingdom is more consistently synergistic, that is interactive, with infection than that of vitamin A. So other things, of course, continued. We know about nuclear receptors, et cetera, for vitamin A. I want to highlight here, since we're talking about sustainable goals and et cetera, that in the 1980s, there was important work that was done by Alfred Sommer and his co-workers in Indonesia and at Johns Hopkins University in the United States, in which they characterized the eyes. Dr. Sommer is an ophthalmologist. They characterized the eyes of children in terms of either being normal, showing night blindness, showing beto spots, which are a classical symptom of vitamin A deficiency, or both. And what they showed was that the the risk of those children succumbing, that is dying at an early age, was actually proportional to the vitamin A deficiency. So the relative rate with night blindness was 2.7 times normal, going up to 8.6 times the normal rate when the children had both night blindness and vitamin A and BTO spots. And so it really led to many great interest and many studies that have since shown that parts of the world that have vitamin A deficiency, as shown by the dark green in this world map, have a high rate, but they also showed that giving vitamin A as a supplement to these children could reduce mortality. All-cause mortality was reduced by about 23%, measles-related mortality by half, and diarrheal disease mortality as well. And so this slide just shows a vitamin A supplementation. Here, these little red capsules contain a high dose of vitamin A that can be delivered directly into the mouth of a child and can be given periodically, like every 4 to 6 months, and provide enough vitamin A to reduce the risk of mortality. Okay, so we can think about this. How? What are the plausible mechanisms? We do know that vitamin A is well known to maintain the integrity of the mucosal membranes and is essential for epithelial cell differentiation. And so we can ask the question, does the vitamin A reduce the incidence of infection? How often infection occurs? Well, the answer to that question is probably no. It's not changing the incidence of infection. But we can also ask, is it a regulator of the immune system? Does it reduce the severity of disease? And the epidemiologic evidence, as well as laboratory data, as I'll show, suggests that this is more the mechanism. And when you think now to our situation with COVID, we're seeing that factors really don't change the rate of infection very much, but they can definitely change the severity of disease and therefore the outcome. Okay, so the answer being yes to this severity of disease question has really led to a focus on vitamin A and the immune response. And so now I'd like to transition to that and to start out with a couple of background facts. So antibodies are the single most effective preventative and therapeutic treatment against infectious disease. Antibodies are really our protectors. All vaccines, that is all licensed vaccines, work by stimulating the body's production of specific antibodies. The antibody response itself is complicated, complex, and it requires multiple what I call handshakes between different cells of the immune system. So first of all, the antigen-presenting cells have to initially recognize the antigen, and they are oftentimes stimulated by what we call adjuvants or immune stimuli. And then the antibody signals are received, and then cells respond. So T cells respond. They interact with the antigen-presenting cells, and they produce cytokines that will stimulate immune response. And then the B cells are interactive with the T cells, and the B cells are the antibody producers. So we need those B cells in order to have a robust immune response. So that really leads to the work that we've done in my laboratory on vitamin A and immunization using animal models for our studies. And my laboratory has studied a number of different antibody responses, but especially has used tetanus toxoid, tetanus, as a model of T-cell dependent antibody response. We also studied the interaction of nutrition, that is vitamin A, and other adjuvants, immunostimulators that augment the initiation of the antibody response. So I'll show you briefly one study and some results out of a number that we have conducted. And so in this study, we have our young animal. These were mice, but sometimes they're rats. And on day 1 here, or day 0, we immunized the animals with tetanus toxoid, and we treated them either with or without an immune stimulator, which is called poly-IC, PIC, and with retinoic acid as the active metabolite of vitamin A. And then 10 days later, we collected blood to measure the primary antibody response. On day 35, we immunized them again. This is the booster dose. But please notice that we did not treat the animals again at the second booster, only at the first. And then we collected blood again. And we measured antibody responses by different methods. So here is a conceptual framework or model of what we predict will happen. You see here at the top, here's our animal that's been immunized with tetanus toxoid. Here are the antigen-presenting cells that are responding to the antigen, interacting with naive T cells, which are going to become activated T cells and are going to produce cytokines. Those cytokines and cells will go to a specialized compartment of the body called the germinal center in the spleen, lymph nodes, etc. And in this germinal center, the activated T cells and B cells, the antibody producing cells, will interact. The B cells will become mature and will produce the antibodies, the IgM initially and the IgG antibodies as shown here, as well as immune memory. So just one slide each for some of these compartments. So first of all, the cells, secondly, the cytokines, thirdly, the germinal center, and then fourthly, the antibodies. So in terms of the cells and what we call co-stimulatory molecules, we found that retinoic acid had a significant effect on a number of these, on natural killer T cells, on various markers that are present that are required for the initiation of the antibody response. Our immune stimulator called poly-IC also had significant effects, but you'll notice they were on different populations. For example, CD8 T cells, NK cells, and a different co-stimulatory molecule. And so these data told us that the two factors, our retinoic acid and our poly-IC, were doing different things, but they might be complementary to each other. When we looked at the cytokines instead, we found that retinoic acid tended to suppress the production of cytokines that are thought to be mostly inflammatory, but it stimulated the production of other cytokines like interleukin 4 and sometimes 10. What we also found was that there was some interaction between the retinoic acid and the poly-IC. For example, retinoic acid suppressed interleukin 12 mRNA and also suppressed the increase, which was due to the poly-IC. So somewhat complex. When we looked at the germinal centers using immunohistochemistry, we can see that the germinal center is staining for PNA, which shows activation of antibody response. Retinoic acid tended to increase that slightly, poly-IC more significantly, and the combination was the greatest of all of these. So the formation of the germinal centers also seems to be promoted by this combination. And then finally, when we looked at the antibody response, we see here the primary response, not much difference due to retinoic acid, but the poly-IC provoked a stronger response and the combination slightly greater. Whenever we look at the secondary response, notice that these levels are much higher. Now we see that retinoic acid, even though it was given much earlier at the time of priming, the first exposure, was significantly higher than the control, poly-IC much greater, and the combination the highest of all. So then going back to our framework model, we came to the conclusions here that there are early alterations in cells and in co-stimulatory molecules like CD80 and CD86, that there is an increase and a rebalancing of the cytokines produced by the activated T cells, that there is maturation and an increased size of the germinal centers in which the B cells mature and become the antibody producing cells, and that there was an increase and a different balance in the primary and secondary antibody responses. Okay, so in terms of concluding comments, I would like to say, first of all, that really every infectious agent is unique. So it's necessary in public health to understand individual responses, individual types of immune agents, because they all have slightly different characteristics. Antibody responses are shaped by the pathogen as well as by the host. And vitamin A as a regulator of cell differentiation is important at several steps in the antibody response, as I've tried to show you very briefly this day. Finally, before I conclude, I'd just like to mention that although none of our studies have involved COVID-19, there's a very interesting review published by Charles Stevenson and George Leitz just this year, in which they compared the effects of vitamin A on other viruses, for example, on measles virus, and tried to relate that to what might be predicted for the COVID virus. And so their paper nicely points out that there can be systemic effects of vitamin A on the intake, absorption, and utilization of the vitamin, such as in Dr. Lee's presentation, on lung tissue, which is also very interesting, and of course where the virus is attaching, on various factors that result in a cytokine storm that may be part of the severity of the disease, and in other forms of tissue damage as shown here. So although we don't have specific data yet about vitamin A and COVID morbidity and mortality, we do have a kind of framework from which we can work based on what is known about vitamin A in other bacterial and viral infections. So with that, I'd like to show you some of the people who have worked in the lab over the years. Dr. Yamamoto is not in this picture, but he definitely was one of them. And we really appreciated his work. And then I would like to thank you for your kind invitation and be happy to answer questions or take discussion.
Thank you, Dr. Ross. ありがとうございました。Any questions from the floor? 日本語でもし質問があれば、私の方でトランスレートしますので、日本語で、でも構いませんよ。So, can I ask one question?
Surely.
Yep. You were showing that the TT immunization and the final picture that vitamin A or the retinoic acid was kind of responsible for not the initial, but rather the late stage of the immune system. You were giving retinoic acid to the animals, but what will happen if you give retinol to them?
Yeah, so we have done those experiments where we have substituted retinol back to the animals, and you get a very similar picture. We like to use retinoic acid because it's really the active metabolite and doesn't result in storage, so it's a little bit cleaner for experimental purposes. But of course, in the public health world, it would be vitamin A in terms of retinol or carotene that would be given. I do want to point out that we were giving the treatment really at the very initial time, the time of priming, and it has a carryover effect to the later response, but we're not giving the treatment again. So we think that initial exposure, the conditions at the time of the initial exposure are really setting the stage for the subsequent reaction. The priming is all important. The boosting is also important, but the boosting just really adds to the stage that has already been set.
So do you think if in the future we should give kind of retinoic acid to the child and then they will have a good immune system?
Well, I'm not going to propose that we should be giving retinoic acid. I mean, I think it would take a lot more preclinical work to do that. But making sure that the child is vitamin A adequate at the time of immunization, I think, is important. And in many of the studies that have been done, that initial assessment really hasn't been done. It hasn't been determined. Whereas we know from really a number of different studies that the baseline situation is really very important for the response. So I think that more public health studies should be measuring baseline indicators and then treating, and then we'll have a much better interpretation of what's going on than if we just assume that populations are homogeneous. I mean, going back to Carmen's study with the heart failure, really trying to parse out who are the individuals who are susceptible through nutritional deficiencies or other conditions, and treating them rather than the whole population is the direction that I think that our research has to go. It's along the lines of precision nutrition.
Yes, I agree. Thank you. We have a question from Tomo Sensei. Tomo Matsura-san. Yeah, I suppose retinoic acid has receptor in the bloodstream, and the complex goes into the nuclear to upregulate genes for immunity. Is this right? If so, what kind of genes are upregulated by retinoic acid? Was the question.
So the retinoic acid that we're giving will be taken up into tissues and will interact with nuclear retinoic acid receptors. Some of the genes that we have observed, for example, we've seen an increase in what's called the activation-induced deaminase, which is important for class switching of antibodies from IgM to IgG or other isotypes. We've seen that in our studies. We have seen increases in certain co-stimulatory molecules that might be important for that initial reception of the antigen. So I think it's not a single gene, it's probably a collection of several different genes whose regulation by retinoic acid makes a difference in the long-term antibody response. Thank you.
Thank you. Azumi Hidas, it's in Japanese. Okay, the vitamin A deficiency is a worldwide health issue right now. What do you think we should do? What kind of treatment can we think to prevent or to I'm sorry, I don't get the right English, but to prevent this health issue, what kind of approach can we think of right now?
So I think I'm just going back to my slide. So I think the approach that we want to be thinking about is to improve nutritional status through food, through agriculture, through better distribution of foods that are appropriate for young children, for example. And I think Dr. Kodish's presentation will address some of that. The strategy that has been used most often up to now is the one shown here. It's really the vitamin A supplementation by capsule. And I think that this has been effective and productive, but there's also a question about, is it sustainable, and is it really the most physiologically relevant way of treating? Probably not. So hopefully we will move away from this kind of supplementation strategy to one which relies more on food fortification or on foods that have been chosen, adapted, and adopted to provide better nutrition. And I think that that's a rather well accepted idea now for not just for vitamin A, but for other micronutrients around the world.
Thank you. Any other questions? 日本語でも私なんとか訳せそうですので。はい。So I have the same thinking that we should switch from supplementations to more like food materials. In Nodai, related to the iron deficiency is also a big problem in Japan. And in Nodai, we made this rice, which has high contents of iron. And we think by giving these irons, we can maybe prevent the iron deficiency of, especially for the women's, it's a big problem right now. So maybe we can make some like vitamin A supplemented like carrots or maybe potatoes or something like that. So that may be one approach that the agriculture university like Tokyo Nodai can do. And yeah, that's what my comments. Any other comments from from Dr. Rossi?
I'm just curious, when you supplement or you enrich the rice with iron, does it change the taste or other characteristics? Does it make it less acceptable?
Yes, the taste. We asked some of the people who are involved and they taste it, and it's not that good. It has the iron has its typical like taste, and so we have to do a little bit more changes in the rice, but yes.
Or some new recipes.
Yes. We have a question from Miyuki Suzawa. I think she can speak English.
Hello, thank it's okay. Can you Can you hear me?
Yes,
I
can.
I think
Hello?
you can hear. Yeah.
Can you hear me? Sorry, it kind of stuck.
じゃあみゆきさん、チャットでお願いできます？そしたら読み上げる。Okay, there was a comment from Stefan. Have you found that the fortified rice is acceptable? And not surely. My answer is it wasn't. We know the Japanese rice, they make real good rice. The sticky Japanese rices, and it tastes very sweet. And so compared to that, and by adding or the fortified iron rice doesn't really taste like that good is the comment, the answer or the comment. Yes. 須澤さんどうですか？メッセージかなんかで来れます？ Miyuki Suzawa is from California. I thought she's one of the alumni of our universities. Well, we can ask another question later. And any other question from you?
Thank you for your talk, Dr. Ross. And so I have a question. I couldn't understand. I'm not an immunologist. So for the last slide you showed us, it was about retinoic acid or vitamin A and COVID-19. that mean? I couldn't get the whole picture of your slides. That means that vitamin A is effective to COVID-19 against COVID. What is
I think the interpretation would be that vitamin A status is important for a good response against the virus, not to suggest that vitamin A is a therapeutic treatment. I don't want to make that suggestion, but we know that COVID-19 attaches to lung epithelial cells, and there's abundant data that vitamin A in the form of retinoic acid is essential for maintaining a healthy epithelium. So tight junctions, normal cells. So I think that the barrier function of the lungs, of the intestine, et cetera, are very dependent upon adequate vitamin A status. So please don't interpret that vitamin A is therapeutic, but rather that adequate vitamin A status is really important for a healthy response to be able to fight the infection.
Okay. Yeah. Thank you very much. Is there any question? Maybe we didn't have a first speaker, so why don't we stop here at this moment and go to coffee break first? Is it okay, Dr. Ross?
Yes, and come back when?
Yeah, come back at like 10 minutes later. It's gonna be like 10 o'clock, what? Okay. 10 minutes later. Yes, 10:15 or 10:15. Is it okay? So we'll come back doing the seminar again at 10:15. So thank you, Dr. Ross, for your great speech, and we'll come back later on. Thank you.
みゆきさんどうぞ。
Hello, Dr. Ross. I'm Miyuki Suzawa, who is from the University of Virginia now. And thank you so much, such a great talk. And I also was interested in the third party, some deficient of the vitamin A in the kids. And I'm assuming it's maybe your study is the third undeveloped country. It's not only the vitamin A deficiency, maybe around the environment, it's maybe some nutrient or some infection disease or something combining of some disease, I'm assuming. Is that why you are focusing only the vitamin A because of the eye phenotype?
Yes. Yeah, well, so your first point, I think, agree with entirely that, in the natural setting, multiple forms. What I think was remarkable about the studies with vitamin A supplementation is that vitamin A was given in the presence of what might have been iron deficiency or protein malnutrition, et cetera, and it still had an impact. So it was really the micronutrient could have a significant impact on survival. And that had a great impact on the public health community. That's not to say that the focus of remediation should be exclusively on vitamin A, but it does show, I think, the impact that a single
in some type of our third undeveloped country is specifically some type of the food based on the culture, the food it's specifically vitamin A missing, or some depend on food cultures, a Western diet or Asian diet different, but some undeveloped country is
it's right. It's very interesting. It's something that I don't think deficiency is still a public health problem. There are abundant food sources of vitamin A, but they're just not used for child feeding. Like in Indonesia, they have fruits and vegetables, but they're not consumed by the children, or they're other health conditions, as you said, sanitation, etc., that are limiting the use. I didn't talk this evening about vitamin A and diarrheal disease, but there's quite an abundant public health and experimental literature showing that vitamin A status is important to enteric infections. Yeah, so I think you're correct. It's a bigger picture, but vitamin A by itself does have a significant impact, as shown in a number of studies.
So,
Okay.
can
Thank you so
I
much.
move on to the second session? Sorry for interrupting you. Okay, so it's time to start. And for the first half of the session, I apologize for the inconvenience of our student talk. So we would like to begin our student talk first and then continue to the next speakers. Okay, the title of our student is "What's so interesting about our research?" Kosuke Sakaguchi and Ayane Arinaga from my laboratory is talking. Please.
Thank you. We apologize for the inconvenience. We are starting over again. My name is Ayane.
My name is Kosuke. We are graduate students in the Department of Nutritional Science and Food Safety. In this presentation, I would like to introduce department and our research with the video. Please watch the video.
I would like to introduce the Department of Nutritional Science and Food Safety. Here we studied safety and function of food. At the University of Agriculture, we are working on the SDGs, and in our lab, we are working on these two goals.
Next, I would like to talk about our research. It's very interesting, so listen up.
What's your favorite food? I like ramen. How about you?
I love sushi. Oily tuna is very delicious, isn't it? Taste is felt by the taste cells in the mouth, right?
That's right. Actually, did you know that there are taste cells not only in the mouth, but also in the stomach and small intestine?
Of course, there are tact cells that eliminate parasites and endocrine cells that secrete hormones, right?
Yes, we are studying the function of these cells called taste-like cells. But there's just one problem. These taste-like cells are very few in number, only a few percent in the body. That's why it's so difficult to analyze and why its detailed functions have not been revealed.
How do you analyze it? That's what we're wondering. How do we investigate the rare cells?
The answer is organoid. We are challenging the functional analysis of these taft cells and enteroendocrine cells using organoid cultures. Organoids are called mini-organs because the culture cells reflect in the body. We are using organoids to increase the number of taste-like cells for our research.
Furthermore, there is one key point in our research. We are generating organoids from monkeys' tissue. In general, most experiments are done on mice. But mice and humans have different taste sensitivities. Therefore, we are generating organoids from monkeys similar to humans, to study the biological defense function and hormone secretion functions of taste-like cells. Actually, what kind of research are you doing?
I'm studying enteroendocrine cells, which are taste-like cells in the small intestine. Now, I succeeded to increase the number of endocrine cells in the small intestinal organoids. I'm able to analyze their functions. My goal is to analyze the function of hormone secretion from endocrine cells. Endocrine cells produce many kinds of hormones, which control our biological function. For example, there is one hormone that promotes insulin secretion and lowers blood sugar levels. If the secretion of these hormones can be controlled by food factors, I think that in the future, we can maintain blood sugar levels without drugs.
Your research is fascinating, but my research is as good as yours. I'm focusing on the pancreatic duct, which connects to the duodenum. The pancreatic duct carries digestive enzymes produced by the pancreas to the duodenum. There are duct cells here that are involved in biological defense.
That's great. What are the features?
Actually, its function remains unclear, so I've established a culture system of duct cells to reveal the function. The duodenum, which connects to the pancreatic duct, has dangerous substances with food. Therefore, there is a risk of dangerous substances entering the pancreatic duct. I'm thinking that there is a biological defense mechanism that senses and eliminates dangerous substances that duct cells have invaded.
That's great. If this is true, it could lead to discovery of food ingredients and therapeutics that activate the pancreatic duct biological defense mechanisms.
Exactly.
Using the organoid culture system, we can analyze cell function in vitro that were previously difficult to analyze in vivo. This technology will help us to develop new functions of food and therapeutics.
Yes, our research has just begun, so we have a long way to go, but we'll do our best to build up. This innovative new technology brings health and well-being to all.
Thank you. That's all.
Thank you, Ayane and Kosuke. So we are going to accept one question from audience. There's no question. 面白い. Someone said, thank you, Stefan.
Thank you.
Okay, so we are running out of time. So thank you very much. We are moving on to next program. Next speaker is Satoko Akiyama, Tomoko Tamura from our university. The title of the talks are A Strong Bond Was Formed from the National Lockdown by Satoko. Please, Satoko.
Okay. Thank you for your introduction. I'm pleased to have this opportunity to speak to you today. Before I start, I'd like to thank for Professor Yuji Yamamoto and Professor Ken Iwatsuki for inviting me to speak today. It's a pleasure to be here. Let's start. Can you see my presentation? Okay.
Yes.
Okay. Thank you. Today, I want to talk about what I experienced in UK. The national lockdown felt like a downfall of rain, but resulting bond like a foreshine rainbow after the rain. It is like the one in the photo I saw from my flat in the UK. I will talk about 4 topics in today's presentation. I'd like to start with part 1, the introduction of the University of Reading. Here, I was conducting research at the University of Reading. Reading is located 60 kilometers west of London and takes 25 minutes by train to London. The University of Reading was founded in 1892 as University College Reading. It has 50 academic schools and departments. The student body of the school is about 15,000. They signed an overseas agreement with Nodai in 2013. Next, I'd like to talk about my life during and after the national lockdown. Here, you can see data of positive COVID-19 cases and deaths from January 2020 to November 2021. The green box in the background represents the time I spent in the UK. National lockdowns and any behavioral restrictions are shown in red. We were only allowed to go out during the lockdown to shop for daily necessities and basic exercise. If you were caught breaking any rules, you had to pay a fine. The behavioral educations after lockdowns were still quite harsh, but they were gradually relaxed and all legal restrictions were lifted on July 21st. So my experience started in the second lockdown, which was a hot topic in the Japanese news. I couldn't even go to the lab during lockdowns due to strict behavioral restrictions. Every day, I would go to the supermarket, take a 30-minute walk in the park, and have online meetings and discussions. I also taught Nodai students graduation thesis and master thesis. And when instructed, I took a PCR test. This was conducted on the university car park. I met the lab members for the first time 6 months after I entered the UK. I was allowed to eat with others. I made Japanese food for many of my friends. I don't remember how much sushi I made. Next, let me begin by speaking about my study. Here, I will talk about my research. Humans eat cooked foods. Different people require different amount of nutrients and functional ingredients. There are many ways to make the same dish. The amount of nutrients and functional ingredients in food changes depending on how it's cooked. The objective of my research is to find a recipe that is suitable for each individual so that all people are healthy. I made pickles using ginger and measured the amount of total polyphenols and polyphenol oxidase activity contained in them. As a result, the amount of polyphenols contained in pickles is low. However, it was clarified that ginger polyphenols remain in foods even when boiled and soaked in vinegar with sugar. Enzyme activity was modest. Further research will be continued with the University of Reading in the future. Finally, I'd like to talk about my impressions in finishing my stay in the UK for a year. There are some things I noticed over the long lockdown. There are things in the world that cannot be changed by your own efforts. It is important to find something positive in any given environment and be absorbed in it. If you accept the other people without having too much rules, the other person will care about you. I have been blessed with good encounters, whether it's a new friend or a stranger. Overcoming a painful event together created a strong bond. It's like comrade. By the way, NODAI accepted short-term international students from France 15 years ago. At the time, I was NODAI student. I reconnected in York, England, with an old friend. Our friendship created by Vice President Mariko Uehara is also one of the bonds. Now, it's my turn to give students a chance to make similar friends. My overseas research was supported by NODAI and also the staff of Department of Nutritional Science who were gracious enough to send me. In the UK, Professor Jeremy Spencer, who welcomed me while he was busy responding to changing country and university rules. Finally, my new friends I met in the UK, they helped me a lot, so I didn't get in trouble. It was supported and realized. That covered everything. I hope you have found that interesting. That's all I have to say. Thank you very much.
Thank you very much for interesting talk. We have to move on.
Yes.
So thank you. So if anyone has comments, please write in the chat, okay? The next speaker is Tomoko Tamura, and her talk is My 365 Days with Rice in University of California at Davis. Please, Tomoko.
Thank you very much. Okay, let me start. So I appreciate the introduction. Thank you for giving me the opportunity to talk about my overseas program. I'm an associate professor of the Department of Food Safety and Nutritional Science. Is that okay? Are there any problem? I will continue. Thank you very much. So I came back here in Tokyo 3 months ago. I went to California University in Davis and studied on rice and diseases. I'm a new face in the field, but the theme helps me to focus on goal 2 on SDGs and hunger and hunger and sustainable agriculture. Those are today's topics. I will talk about not only my study, but also my life in Davis. On housing. Before going to the US, I subleased an apartment. Since my sabbatical period was only 1 year, I wanted to settle down as soon as possible and focus on study in my host lab. This is a picture I took from an airplane just before landing at California. They have smoky days in summer since wildfires happen. The rate of wearing masks in California is so high. Inside of the red line is Davis. It's surrounded by a field, so a little bit countryside, but so safe. As for weather, summer starts in April and low humidity, but super hot. The map shows forecast on Thursday in June. So 38 on Wednesday, 43, 42, 39, 38. So don't you think it's cold on Monday? Squirrels are melting because of those crazy heat waves. As for people in Davis, they are friendly and kind, pets as well. The smoky day I arrived at the airport, the first English I heard is "Welcome to Davis." On an airport bus, a man smiled at me and said so. We talked a lot then. So everyone knows coming foreigners can expand coronavirus crisis. I'm not sure his real feeling, but I think citizens are considerable and people's feeling have a big heart and global sight. As for food, fruits and vegetables are fresh and delicious. Those are vegetarian, vegan, and pescatarian friendly ingredients and dishes. My image is of food in America, such as oily hamburgers and pizza, French fries were completely wrong. Let's move on to the campus in UC Davis. It's huge, you need a bike. On COVID test, all people living in Davis can take PCR test once a week for free. We can get the result on our smartphone quickly. No reason to skip going to work. There were about 10 members in the lab, and we are international because American, Argentina, Brazilian, Chinese, Indian, Korean, Mexican, Taiwanese, and only Japanese, which would be me, were in the same lab. So despite our having a different culture, we keep a good order. We communicate well. Guess what do you want is one of our motto here in Japan, but not the #1 slogan in this situation. This is a way to get to the lab. I walked to work. This is a building of plant pathology. Let's go into the building. She's Professor Ronald, who is a plant pathologist and geneticist. She gave me enough space to perform experiments. Let me talk about our team study. Those yellow leaves are infected by Xanthomonas. It's like a bacteria. Plant diseases are major causes of crop yield losses. RAX-X is a small sulfated peptide and is secreted by a Xanthomonas bacteria when it infects rice. And XUA21 is the receptor isolated in the lab and mediate immune response in plants. RAX-X mimics the sequence of PSY1 expressing in plants and promoting cell growth. Now, the lab focuses on genetic components controlling rice pathogen interactions, structure activity relationships between the ligand and the receptor. We haven't isolated the receptor of PSY1, and I focused on the family of PSY1. In rice, PSY1 has a subfamily. We created ubiquitin PSYs overexpression lines in each member of the subfamily. So actually, I used to go to the greenhouse and grow lots of rice to get the homozygous lines. Using the OX lines, we've examined the phenotypes of the OX lines and whether the OX lines modify the immune response or not. Our curiosity is also, is PSY peptide secreted from plants? And is PSY secreted from plants received by other plants? So those are ongoing projects. So let me skip the results. Sometimes we had lunch parties at the backyard of the lab building and downtown in Davis. We went to San Francisco, Lake Tahoe. There I got a car sickness, and you know why I'm holding the plastic bag in my mouth, right? My private life is also full of smile. I taught origami paper crafting to children living in Davis. The other day, my Brazilian friend played violin. I enjoyed playing street piano. I appreciate that Nodai gave me the chance. I appreciate that UC Davis and the lab allowed me to study. My talk is a small part of my experience. I did what I want to do. I tried what I was able to do. I was a daring explorer. Then I had amazing days. I'd recommend my students to study abroad. I wish them to become people who has global sights and big hearts, like a man who said to me, "Welcome to Davis," when I arrived there. Thank you.
So we are going to accept the question by chat, and we are running out of time. We will move on to the next program. The next program is short lectures from Penn State University. The first speaker is Dr. Gregory Scherer, a professor of nutrition and physiology. His talk is Oxylipids, a molecular mechanism for regulation of intercellular signaling by dietary PUFA intake. So Greg, please.
Thank you so much. And I just wanted to let Dr. Tamura know that I know a lot of those places that you were talking about because I went to UC Davis. But I wanted to thank Dr. Yamamoto and Dr. Iwatsuki for the opportunity to speak to this audience. And it's just, it's really exciting to get to talk to a group of people who are interested in a lot of the same things that we're interested in here at Penn State. So I just wanted to start by just talking a little bit about why we think polyunsaturated fatty acids matter. Polyunsaturated fatty acids are not just energy sources. And I want to talk, I'm going to focus on a particular polyunsaturated fatty acid called arachidonate. And I'm going to use that as a jumping off point for describing why polyunsaturated fatty acids as a category are important, and then talk about why individual and specific polyunsaturated fatty acids matter. But in our lab, we see at the end point of polyunsaturated fatty acids as regulatory substrates for signals called oxylipids. And before I just describe what oxylipids are, and I'll try to be brief because I know we're short in time, but I want to just really quickly remind you guys there are a lot of problems with our understanding of polyunsaturated fatty acid signaling. And this plays out in a lot of the interventional research we do to try and treat diseases. Recently, there's been a lot of conflicting information in randomized controlled trials when it comes to omega-3 fatty acids as an interventional strategy. And I'll just say that EPA and DHA as a treatment for prevention of myocardial infarction has had some big hits and some really null results. And so that's been a source of a lot of confusion. Also, HDL lowering therapy has been very confusing in that there have been a number of strategies to raise HDL. I put HDL lowering, I meant HDL raising, but the point being is that interventional strategies to raise HDL as a therapeutic for cardiovascular disease has not worked. And there's been a number of challenges in other areas as well. So I just want to talk to you guys a little bit just to remind you where the convergence of polyunsaturated fatty acids are, how they relate to plasma lipids like lipoproteins. And that is because it's in lipoproteins that polyunsaturated fatty acids are transported. And just as a reminder here, I put this slide up here. Forward transport of lipids, and you know, these are commonly thought of as cholesterol, but all lipids are transported in a forward direction in LDL, IDL, VLDL, or chylomicrons, and there's various mixtures of triglycerides and cholesterols in these lipoproteins. But HDL is unique in that HDL is involved in reverse transport of lipids, that is from peripheral tissues to the liver. And so I want to go through where plasma oxylipids are located before, and then I'll tell you guys exactly how the heck these oxylipids are produced. But the point here is that here is a particular oxylipid. It's called 18-HEAP. It's produced from eicosapentaenoic acid, which is an omega-3 fatty acid, commonly known as EPA. And you can see here its levels are very low in when it circulates bound albumin, when it circulates in LDL, or when it circulates in VLDL. But in HDL, very high levels, whether we have an animal in its basal condition or under some sort of metabolic stress. In the case of this experiment, these were animals that had a TAC surgery, which is just a way of inducing heart failure. So whether in metabolic adaptive stress or in the basal condition, 18-HEAP was where we found most of the vast majority of these of this oxylipid circulating. Here's a different one. This one is made from linoleic acid. It's called 13-HODE, and it's produced from an 18-carbon fatty acid. And I think for those of you guys who are involved with studying plants, linoleic and alpha-linolenic acid are the primary polyunsaturated fatty acids produced by plants. And I think it's it's really underappreciated that these polyunsaturated fatty acids are directly used to produce signaling metabolites like oxylipids. And so, just throwing that out there for you guys to think about. But you can see here under basal conditions, in particular, HDL is where we see this particular oxylipid, and not so much under metabolic stress. And the third one I'll show you guys is 12 heat. This is an interesting one, and I'll focus on this one for the duration of what I talk about. 12 heat, again, under conditions of metabolic stress uniquely is where we find it. So when the animal is given this thoracic aortic constriction surgery, which induces a stress on the heart, we see it begin to appear in HDL. So here we see this convergence of lipoprotein metabolism and polyunsaturated fatty acid metabolism. And so we wondered how oxylipins could appear in HDL. The first thing to recognize about oxylipins is they're produced inside the cell. And the source of transferring oxylipins from the cell into HDL is ABCA1, and particularly ABCA1 and the ApoA1 axis, ApoA1 being the primary structural apolipoprotein of HDL. So as I promised, I would tell you a little bit about how oxylipids are produced, and this is a pathway, a signaling pathway of polyunsaturated fatty acid metabolism. And here in this lipid bilayer, we see in black, or this black, yellow, and blue complex is a single phospholipid, and within that phospholipid, there's a hydrophilic head and a hydrophobic tails comprised of two fatty acids. One of those fatty acids is typically a saturated fatty acid, and the other is a polyunsaturated fatty acid. Here in red, we have the specific phospholipids that are arachidonate, and they are released by activation of an enzyme called phospholipase A2 to release arachidonate into the cytoplasm, and a number of different enzymatic pathways. You're probably most familiar with cyclooxygenase. That's what we take aspirin for. It produces prostaglandins via the action of an enzyme called cyclooxygenase, and those are associated with activation of inflammation. But cytochrome P450 epoxygenase can produce potent anti-inflammatory metabolites. 12 and 15 LOX produce 15 and 12 heat, which have these mixed inflammatory results. And I showed you just earlier the production of this particular one, and this one ends up in HDL. And as well as there's other autooxidative pathways that I won't talk about. And so to kind of describe this signaling process, we have here activation of phospholipase A2, the production of arachidonate. In the case of 12 heat is important because it converts via the action of 12-lipoxygenase. It mediates the production of a cytokine many of you guys are probably familiar with called MCP1. And so just to make a comment, Dr. Ross was talking about a lot of the implications to COVID. Many cytokines, their production inside the cell is mediated by these regulatory oxylipins and their production inside the cell. In this case, 12 heat inducing the production of MCP1. And there's a lot of discussion, is that quote cytokine storm in COVID reflective of a storm of oxylipids inside the cell? Well, inactivating that 12 heat inside the cell, you at some point you're going to want to turn MCP1 off, and so we can turn that off by resequestering the 12 heat inside the phospholipid bilayer. So just like it was arachidonate when it was released, it's converted to 12 heat, and then it's resequestered in the plasma membrane. And we hypothesized that the way it wound up in HDL was because ABCA1 combined with ApoA1, so dependent upon both of these proteins, mediated its transport into HDL. And we thought that that would serve the purpose of preventing recycling of this 12 heat via subsequent action of phospholipase HG. And so we just did some, Yachi Lee was discussing tracer experiments. We did a compartmental model using tracer lipids, just like she did with vitamin A. And we created a kinetic model. For the sake of time, I won't get into it. But we did an experiment where we added those two proteins that we said the efflux of those oxylipids would be dependent upon. So we either added or not ApoA1 or vehicle to a cell culture of just raw to 64.7 macrophages, a very generic type of macrophage that you can grow in the lab. And we let ABCA1 be expressed. We induced the expression of ABC1, or we silenced it using siRNA. And again, for the sake of time, I won't go too much into the experiment, except to say we developed this a kinetic model where we could trace the cell arachidonate, the cytosolic 12 heat, the amount of 12 heat in that plasma membrane, and its efflux into the media, either as 12 heat in HDL or as free 12 heat. So I'll just show you guys some of, briefly, I won't show you all the results that we have, except to say that this was the basal condition where we didn't add any exogenous ApoA1, but we allowed ABCA1 to be expressed in the cell. And under these conditions, I just have them all color-coded here. We're not showing arachidonate, but you can see that reasonably high levels in the 12 heat that accumulate to reach a steady state inside the plasma of the cell. And it accumulates within the membrane of the cell, but there's no export there's in fact no detectable levels of heat either in the media or non-esterified. So without ApoA1, esterified oxylipids do not accumulate in the media. But when we add exogenous ApoA1, the first thing that happens is you'll notice that the amount of 12 heat in the cytoplasm drops precipitously. And we still see accumulation of 12 heat in the plasma membrane, but importantly, we begin to see the accumulation of that 12 heat in HDL. So by activating the accumulation of 12 heat in the HDL, we see a concurrent drop in 12 heat in the cytosol. So when this compartment drops, it goes up in that compartment. When we silence the enzyme that's responsible for exporting, we again see a high level of cytosolic 12 heat, and we see low levels or undetectable levels of 12 heat in HDL. But we do begin to see an accumulation of non-esterified or just general efflux of 12 heat. And I won't show you the rest again for the sake of time, but just wanted to kind of show you that why we see that as being important. So the net flux, the fractional transfer of 12 heat is really only present in high levels in the condition where we have both ABCA1 being expressed and ApoA1 present to mediate this transfer from the cell membrane to the HDL. And the result is, again, that we see a great reduction in the amount of cytosolic 12 heat. And just to show you guys, it's this compartment that's thought to be the active signaling compartment. And we see exactly that the cells with the lowest levels of 12 heat have the highest levels of MCP1. And so when you completely take apart this export axis, you have very high levels, high rates of MCP1 accumulation in the media, which would be analogous to MCP1 export by cells. And as we rebuild and we create the mechanisms for exporting that 12 heat to HDL, the rate of MCP1 accumulation and the maximal amount of MCP1 accumulation in the media drops. So we see this as a promising way to describe the ways in which polyunsaturated fatty acids are important for regulation of inflammatory mediators, but also how they co-participate with the function of lipoproteins in order to accomplish this regulation. So in summary, there's a strong dependence of oxylipid appearance in the media in the HDL on the presence of ABCA1 and the availability of ApoA1 to accept those esterified lipids. And the effect of HDL dependent efflux is to lower intracellular 12 heat and reduce macrophage MCP1 production. And lastly, I just want to just tell you why different types of fatty acids are important. That's just a description of the role polyunsaturated fatty acids play. But it's important to remember that that's just a single 12-LOX metabolite of arachidonate. And in fact, when you use EPA, you get another product called 12-HEAT. And so here is that signaling pathway based on arachidonic acid that I showed you, where arachidonate is used for all these different production of all these different signaling metabolites. When you substitute that metabolite, that substrate for EPA, you produce an entirely different set of oxylipids, again, each with their own unique metabolites. And so we see this as a promising way to describe why individual polyunsaturated fatty acids are important and how they play a role working together. These are the people in my lab. I've got in boldface the people who are active in my lab, including this guy who did much of the work that I showed you today, and Carmen, who showed you her work in heart failure and its relationship to DHA. So thank you very much, and I'll take any questions if there's time.
Yes, thank you, Greg. Interesting talk. Are there any questions from the audience? I have one. Can you read? Because we are familiar with your study, I would like to ask whether oxylipin is an anti-inflammatory or not.
Yeah, and I think that's a great question. And so the answer is yes to both. So oxylipins are, in this case, in the case of EPA, when EPA is used to make a cytochrome P450 epoxygenase, so an EPEATer, it's anti-inflammatory. But when EPA is used to make 15-HEAT, the answer is it's probably pro-inflammatory. And so these ideas where we assign, say, for example, EPA as being anti-inflammatory and AA as pro-inflammatory are probably generalizably true, but not exactly true. And so the answer is yes, they're both.
Yeah. Okay. Thank you, Greg. Is there any question? If not, thank you very much, Greg. We would like to move on to the last speaker. Okay. The last speaker is Dr. Stepan Kodish, an assistant professor of nutrition and biobehavioral health. His talk is about understanding social and behavioral factors important for child nutrition in global context. So Stefan, please.
Okay, hajimemashite, Stephen Kodish to moshimasu. Yoroshiku onegaishimasu. Let me see. When I was in my early 20s, more than 15 years ago, I went to Japan for a couple of weeks, and I was invited by friends to go to dinner, and I poured soy sauce into my rice, and immediately my friends at dinner said, no, 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 no. And what started as a 3 or 4 week trip turned into 4 years exploring Japan, living in Tokyo, learning about the culture, falling in love with how different socioculturally Japan was from the United States. And I realized that I wanted a job in the future that combined food, that had travel involved, that explored cultures, and that somehow helped to address health and human rights. So my PhD work after Japan was at the Johns Hopkins Bloomberg School of Public Health, where I focused on the social and behavioral sciences, working with a nutritional anthropologist named Joel Gittleson. And then my postdoctoral work was with a nutritional epidemiologist at Harvard School of Public Health. And I like to put those two worlds together in a mixed methods approach to try to understand both what's going on nutritionally at the population level, as well as why those phenomenon are happening at the societal and behavioral levels. And our lab focuses on, just like Tomura-san, trying to end hunger, achieve food security, and improve nutrition by addressing SDG number 2. And I thought this would be such an easy thing to do over a decade ago. But what I learned is that it's a complex situation. We have these basic causes such as economics and food prices, etc. We have these underlying causes such as household food security and the way that people care and feed their children, as well as the health systems and wash-related environments. And then, of course, disease and dietary intake are two of those immediate factors. And I was most interested in dietary intake and the drivers of it. So come with me and we can travel around the world a little bit to look at what some of those diets do look like. Here's an example from the US. This is what the Rivas family eats in just 1 week. You can see a lot of processed foods, high sodium foods, and you might guess this family is susceptible to overweight, obesity, non-communicable diseases, as well as a huge impact on the environment with all the processed and packaging around those food items. Similar in the UK, types of diet and the same types of risk factors for nutrition-related illnesses. Now, if we go to a middle-income country like Mexico, the diet starts to look a little different. We have more fresh fruits and vegetables, but they're probably seasonal in nature. You also see a lot of fizzy drinks, such as Coca-Colas, as this country moves to the nutrition transition. This family certainly is susceptible to overweight and obesity, but also micronutrient deficiencies. Oftentimes in the middle-income countries, we're seeing this double and triple burden of malnutrition, sometimes in the same household. You might have an overweight father and a micronutrient deficient adolescent girl or young child. And the same picture in Turkey, where you see a middle-income family, lots of fresh fruits and vegetables, but you don't see a lot of animal sources in this diet. And so the micronutrient deficiencies manifest not only through vulnerable life stages, but also contribute to chronic undernutrition for young children during that window of opportunity from conception until 2 years of age. Now let's keep traveling, and we'll go past Guatemala, but we'll come to Mali. And Mali is a family that is spending about $25 US per week on their food. And you can see the diet is very different. Very few animal sources, if any, mostly grains that are probably not fortified, a large family size, and young children, adolescent girls, pregnant and lactating women in this family are probably susceptible to acute malnutrition, as well as micronutrient deficiencies. And then this population, or this family, is the type of family that I usually work with and try to improve their nutritional status. This family lives on less than $2 per day. They're in a refugee camp, and you can see how they both are lacking nutrients as well as calories in their diets. I don't see any animal sources and very few grains, very few, if any, vegetables or fruits. This family is certainly susceptible to a range of different nutritional illnesses. So throughout the world, different nutritional illnesses manifest based on, in part, local diets. So let's zoom out, and it's no surprise then that the global nutrition situation looks like this. And you can see where the orange and the dark gray countries are. You'll see triple burdens of malnutrition, where moms or dads might be overweight or obese, but at the same time, anemia and chronic undernutrition are in the same households. And the biggest driver of this is, of course, income. In public health, most public health outcomes are determined by income. And I think it's just important for all of us to keep in mind that there are 1 billion with a B, people living on less than $2 per day. That's today. And that's pretty amazing statistic. Now, in humanitarian nutrition, we have types of specialized nutritious foods. So on the top right, we have micronutrient powders that look like this. On the bottom right, this is a fortified cookie provided by the World Food Program for young kids. And there's a whole range of these specialized nutritious foods. So I was so excited to get into this business because we have a solution, regardless of the life stage, regardless of the type of malnutrition, to fill important nutrient gaps. Well, my first study in 2009 in this field, we were trying to address micronutrient deficiency at Kokuma refugee camp in Kenya. And Kokuma is in the northwest part of Kenya. And in Swahili, Kokuma literally means nowhere. There's nothing around it. It's a protracted refugee setting that's been around since the early 90s as a result of the South Sudanese war. It's a very diverse place with Ethiopian refugees and Congolese refugees and Sudanese and South Sudanese and Somali and Kenyan nationals. This is a picture from Kokuma of a school. This is a Sudanese Dinka family. You can see the large family size, and you can see the steel, the corrugated steel roofs that are more emblematic of a settled type of community rather than a traditional refugee camp. At Kokuma, anemia is prevalent among almost 50% of women of reproductive age, as well as young children. And of course, anemia is largely attributed to either iron deficiency or disease or both. We had been working with the World Food Program, which won the Nobel Prize just a couple of years ago, as well as with Sight and Life, which is a part of DSM. And they had been implementing these micronutrient powder interventions in several different places around the world. And micronutrient powders have 15 to 17 vitamins and minerals that are meant to be fortified into a baby's complementary food or anyone's semi-solid food. So for this intervention, we provided this to everyone in the refugee camp as part of their typical food distribution. This is a food distribution line at Kokuma. So as people move through the line, they would receive the micronutrient powder. These were the scoopers who were handing out both the food and the micronutrient powder. Here is a picture of a school, and we had some promotional posters that were promoting this MNP to the young kids. We had promotional t-shirts that were trying to promote the MNP at little kiosks. And what we found was that after a couple of months, the uptake of the MNP at the distribution center dropped to almost 30%. That means that only 30% of the people going through the line were even taking it. So we invested more money, and you can see that the uptake improved a little bit, but uptake and consumption are not necessarily the same. And we found that over time, about 45% on average of the people were taking, not necessarily using the micronutrient powder. Our impact evaluation found no change in anemia among women, no change in anemia among children, and the overall prevalence of anemia did not change. And this was my first project working in this field, and I was so disappointed. So we did another follow-up study that was mixed methods, and we found that it was a complex situation. If you looked at the left-hand side of the picture, these are all the upstream factors from the implementation planning to the cohort study that we had that were poorly planned and not much resource invested in. As a result, there was a lack of information. There was not enough social marketing or communications to promote and to teach how to use the MNP. And then that led to downstream challenges. People perceived this to be a condom rather than a micronutrient powder. People did not like the logo. They thought it looked like a genie and would not use it. There were also different types of effects on the body. Some were positive, but some that were positive, such as kids' appetites increased, are not a good thing at a refugee camp where each family only gets 2,000 kilocalories per day of food. So a lot of mothers said, well, I don't want my children to be hungrier, so we're going to stop using this. Overall, it was a highly complex behavioral situation with all of these factors that led to the low uptake and usage in summary. And if you think of this, it looks like a simple behavior. Put this into the semi-solid food every day, but actually there are a lot of steps. First, mom has to perceive that there's even a risk of micronutrient deficiency, but a lot of people know that until micronutrient deficiency like anemia gets too bad, you can't even tell if your child is sick. So why would I use this if I can't perceive my child to be ill? Second, you have to have the confidence that this is safe for your young child to use. You have to be able to have the adequate complementary food for it to be fortified in. You have to get firewood to be able to cook. You have to start the fire. You have to get water for boiling the fire to cook the food. Then you have to add the maize flour to create the semi-solid food. And then you have to let the porridge cool enough so that it doesn't break the iron encapsulation before adding the MNP. Then you have to fortify the MNP, but not all of it because babies have such small stomachs that they usually won't eat it all. So just a portion of it to avoid leftovers. You have to have clean utensils and hands before feeding the young child. And then hopefully there's no sharing of the food among other children. And then this has to happen every single day. Wow, that's a very difficult type of behavioral intervention. What seemed so good on paper was actually very difficult in practice. And we know that this is efficacious at the population level, but it's definitely not effective in the implementation of the intervention. So micronutrient powder supplementation, it's a great intervention on paper, but behaviorally, it was very complex. Can I take you to one more place just very quickly? And we can travel to Niger a couple of years later, where we have a different specialized nutritious food. And Niger is in Central Africa. It's one of the poorest countries in the world. And every year, many children suffer from acute malnutrition in Niger. And acute malnutrition of children coincides with the end of the harvest. I'm sorry, just before the new harvest of food, and coincides with the rainy season. near the end of the rainy season when there's puddles and the malaria prevalence or malaria incidence increases. We had this great product that was invented a couple of years ago that has vitamins and minerals. It's called Plumpy Nut. It's an innovative, literally a life-saving breakthrough. It can bring kids back from near death, and it tastes good. It has a peanut butter base because it has both kilocalories and vitamins and minerals. However, we found that in a program coverage survey, only 1 in 4 children were actually utilizing or being fed the Plumpy Nut as a treatment for acute malnutrition. So we flew to Niger to figure out what was going on. And here was our team. At the time, I was with Doctors Without Borders doing a study. This was my bed, and you can see the bed net at the top to keep out the mosquitoes. And this was our team getting ready for field work. And we traveled through very rural Niger. You can see how difficult the agricultural would be in this context. And this was the road infrastructure. It was all dirt. And we saw animals who were not grazing on agriculture, but were grazing on trash, and an important indicator of poverty. We stopped here, which is actually a gas station. So we filled up the car with fuel. And then we got to our community, which looked like this. And we met the community leaders to get approval to work in their community, and they were very intimidating. And we got to see the animals and the different dwellings, and we talked to people. These were kitchens, and you can see there's no ventilation in the kitchen. Acute respiratory illness is a big challenge in global health. We visited health centers that were not always open. We talked to leaders. We met local transport. This was a local person who would take you to the health center if you paid him enough to ride on the camel, if you had a sick baby. We visited malnutrition clinics. We went to weighing stations and reviewed secondary data. We looked at and analyzed that data to try to draw assumptions or conclusions. And we used different anthropological methods to try to understand conceptions of this Plumpy Nut and why it was not being utilized as planned. We found a couple of different things. Number one, this had the opposite problem of micronutrient powder. MNP was not being used. Everyone except the children were eating the Plumpy Nut because it tastes so good. The organoleptic properties in a place where food security is very minimal was being eaten not by sick children, but by dads, brothers, mothers, and even other siblings. Number two, this is a highly interdependent cultural context. And in Niger, you don't give a food item to one child, you give a food item to all children or the entire family. And understanding that context was important for understanding the appropriateness of this intervention modality. And then next, this was being traded a lot as a commodity. It was being sold for other resources. Number two, take a look at these drugs. You have emergency contraceptive. You have finasteride, which is for male pattern baldness. You have Viagra. You have PrEP for HIV prevention. And you have Plumpy. And we found that Plumpy had the same type of symbolism as these other types of products. There was a stigma attached to it. You don't talk about this at the dinner table with your family, do you? Or you maybe don't tell everyone about going to the pharmacy to get some of these products. And Plumpy Nut, in this setting, symbolized being a bad mother or being a poorer household than other households. Or it reflected poorly on the head of the household, the father, because it indicated that maybe he wasn't being able to provide enough resources. So moms were reluctant to use it. And then number three, we found that this was a very biomedical type of approach. A biomedical type of approach to treat malnutrition, or a clinical approach, assumes that pathogens cause disease. And we, in anthropology, say that's part of an internalizing medical belief system. Pathogens in the body can disrupt that balance. But in Niger, traditional medicine practices exist. Biomedicine is not something that's learned in school. where a lot of people don't go to school. And so karma or superstition or other beliefs were actually driving a lot of explanations for sickness. So why, mothers told us, why would I use this if it's not a pathogen or a disruption of my body that's actually causing my young baby to be sick? The young baby might be sick because actually my neighbor did something, or my husband did something to the neighbor 2 years ago, or because it's related to traditional medicine. So our approach, although good on paper, again, was not a perfect fit for this cultural context. And this is the type of work we do here in my lab, but we have all of these products and they seem great from our nutrition perspective, but we need to think more deeply about the sociocultural contexts where we introduce them. Behaviors do not occur in a vacuum, and I think that's important for we nutrition implementers to remember. Thank you to our team, and I welcome any of the NODI students to come visit us and to join in this work that we think is quite important. So thank you very much. Arigatou gozaimasu.
Thank you very much, Stefan. I could accept only one question because we are running on time. What does nutritional powders made from? Are those for vegan? I don't know what kind of powders they are referring to, but do you know, Stefan?
I do not
Which
know
one?
the answer. I don't know whether it is vegan, but these are all synthetic vitamins and minerals. Yeah.
Okay, thank you. So thank you, Stephen, for the great talk, and we appreciate all the speakers. And I have to move on to the concluding remark. Okay, thank you, Stephen. So before closing the symposium, we would like to thank all the audience for participating in this meeting. It was really very early in the morning here in Japan, and late at night in Philadelphia, and more than 120 person have entered this site and watching our symposium. I believe people enjoyed the symposium and could have an idea what's going on in both sides of the world. Tokyo University is celebrating its 130 years anniversary. But I found Penn State has history of 166 years. So we are in the just middle of developing ourselves. So next, I would like to thank Dr. Ross and people from Penn State and from Tokyo University of Agriculture as well. We have learned scientific aspect of Penn State and about us well. I think this is the first step to know each other. And from now on, we could start communication and seek for collaborative work or exchanging our students or faculties. Please do not hesitate to contact us. We hope our bright future based on our great friendship and good research. Thank you, everyone.
ありがとうございました
Thank you. So we are closing now. Thank you for
お疲れ様でした。
participants and audience. Thank you.